you, Kevin, for the nice introduction, and welcome everyone. Sorry for the delay with my computer. So I will talk about the role of X-chromosomal dosage. So, is this working? No. So as you know, females have 2 X chromosomes, while males have 1 X and a Y. So while the Y chromosome is really small, has very few genes, the X chromosome is a big chromosome, has about more than 1,000 genes, and most of these have functions that have nothing to do with sex, so they should work in similar ways in males and females. And therefore, at least for some of these genes on the X chromosome, it appears to be important that they are present actually at a similar dosage in males and females. And to achieve these kind of dosage compensation for X chromosomal genes in mammals, the process of X chromosome inactivation has evolved, where each cell in a female will is more or less completely silence one of its 2 X chromosomes. And this process is random in a way that in the end, half of the cells will inactivate the X chromosome from the mother, and the other half of the cells the X from the father. This process is initiated very early during embryonic development, and then the inactive state of one of the chromosomes will be inherited to all the daughter cells throughout the life of the organism, such that in the end you essentially have each female as a mixture of cells with the paternal or the maternal X chromosome active. So this can be shown quite nicely with these type of cats, where the orange and the dark color is encoded by an X chromosomal gene, such that females can have kind of patches of fur that is orange and patches of fur that is dark, depending on which of the 2 X chromosome is active in these cells. While her brother here can only have one of these 2 colors because he has only 1 X chromosome. So X inactivation is controlled by the master regulator that's called Xist. So this is a long non-coding RNA that is expressed exclusively from the inactive X chromosome. So it's only expressed in cells in female cells, so in cells with 2 X chromosomes, and also here it's only expressed from 1 out of these 2 chromosomes. So while all the other genes on the inactive X are silenced, Xist is still transcribed, and the RNA kind of covers the entire X chromosome and initiates gene silencing. So this is kind of the developmental cycle of mice, where I've indicated the status of the X chromosomes. So very early after fertilization, you have this imprinted form of X inactivation. So that's specific to mice and probably doesn't exist in humans, for example. And here, always the X chromosome from the father will be inactivated. Now, this imprinted X inactivation is then reversed here at the blastocyst stage in the inner cell mass. These are the cells that will give rise to the embryo later on. So here now you have 2 active X chromosomes for a short developmental time window. And just subsequently, the cells will undergo random X inactivation, where each cell will either inactivate the paternal or the maternal X chromosome. So what I will talk about, the project I will talk about, is about the role of having X chromosomal dosage here during development, but I included in the introduction a few slides to show you that maybe X chromosomal dosage might also have effects in somatic tissues. So, and this is mainly because it's actually not all cells on the X chromosome are completely silenced, but there are genes that escape X inactivation, which means they're still expressed from the inactive X chromosomes to some extent, at least. So this is just from a study where that looked at many different tissues in mice, and you see that each tissue there's between 5 to 10% of genes that will escape X inactivation. And so this has been established in a long time that escape, except in one tissue here, they found where there's much more, but in general, escape from X inactivation is quite rare in mouse. This is somewhat different in humans, where quite a number of genes actually escape axon activation. So this was from a recent study where they looked across several tissues quite systematically, I think, where they found that there is, so this is there are about 10% of genes that will escape axon activation in essentially all tissues, but then you also have genes that only escape in a subset of tissues. And this actually does lead to gene expression differences between males and females. So this is a map that was I took from that paper on the human tissues. What you have here along is the entire X chromosome from human tissues, and each column is a gene, and each row is different tissues. And what is plotted here in this heat map is that it's essentially sex bias of gene expression. So if it's red, it's higher expressed in females, while when it's blue, it's higher expressed in males. So we have this region here at the end of the chromosome in the pseudo-autosomal region, where genes are expressed more highly in males, and this is because they have actually homologs on the Y chromosome, these genes. But then in the rest, you see these red stripes, which are essentially genes that are consistently expressed higher in females across all tissues. And so about most of these genes, we don't know really much what is their function, but it's clear that this dosage difference does have some effect on phenotypes in humans in particular. Because there are these different syndromes that are associated with different genotypes. So there are, for example, women that have just a single X chromosome, so they are XO. This is called Turner syndrome. And there's also men that have two X chromosomes, that have essentially XXY, which have the so-called Klinefelter syndrome. So in this case, one of these two X chromosomes will undergo axon activation, but still both of these genotypes will have detectable phenotypes. So both the kind of neurological differences, but also in appearance. There is also X trisomy, for example, so women that have 3 X chromosomes or 4 X chromosomes. So what happens here is that all except 1 X chromosome will be silenced, and these have actually quite mild phenotypes, but they can still be detected. So what I just want to show you now, a few slides on that maybe these genes that escape axon activation or X chromosomal dosage might have some effect on sex bias in susceptibility to different diseases. So I just took this from a review where you get the number of different diseases that have different sex bias, in particular autoimmune diseases. Many of them tend to be more frequent in females, and this is just a more detailed map of the incidence of, for example, you have here lupus or rheumatoid arthritis that are more prevalent in women. So of course this is probably strongly affected by hormonal differences between males and females, because they're not only different with respect to the chromosomes, but also with respect to sexual organs and hormones. But there's some data that indicates that actually also these genetic differences might contribute, because I hope you can see this. So I copied just a table from a study where they looked at the incidence of SLE and this Sjögren's syndrome in people with different X chromosome numbers. And what was found is that people who have a single X chromosome, so which is men, but also these XO Turner women, they have a quite low risk for these diseases. While both women with 2 X chromosomes or men that have 2 X chromosomes, they have about more than 10-fold increase in incidence of these diseases, and this is even higher in women who have 3 X chromosomes. I suggest that also a gene dosage plays a role in this. So we don't really know how this actually contributes, but there is a number of genes on the X chromosome that have something to do with the immune system. This is just a few examples of those. So for example, there's FOXP3 or IL-13 receptor, or these toll-like receptor 7 and 8, for example. And for this toll-like receptor 7 in particular, it has actually been shown that the dosage of this gene can affect like autoimmune diseases, because there is this mouse, kind of a mouse model for lupus where part of the X chromosome has been translocated to the Y, such that TLR7 is present at a higher dose in the male mice in this mouse model. And this leads to increased incidence of autoimmune diseases, meaning that if you have higher levels of TLR7, you're probably more likely to develop autoimmune diseases. So whether now this has any effect on, let's say, normal women, depends obviously on whether TLR7 is completely silenced by axon activation, or is it actually maybe present at a higher dose in females. And there just very recently was shown that it does actually escape axon activation in some immune cells. And this might actually be a bit more global effect, what we don't, I think this is not well understood, but it was found that at least in some subgroup, like here, what is shown here, some stages of B cell development, that there are some, let's say, aberrant profiles with respect to Xist expression. So I'll explain what you see here. So this in these cells, what the stains is Xist. So this is this non-coding RNA that codes the X chromosome. And normally, what you see is this kind of cloud because it covers the whole X chromosome. However, this cloud essentially disappears here at the pro-B cell stage, where it's not detected anymore. It comes back maybe in a weak way later on during differentiation. But this could mean that axon activation is somewhat loosened in these cells. Also, it doesn't mean that the X chromosome will become active here, because there's a lot of other mechanisms that will that repress X chromosomal genes. Okay, so this was my kind of detour to role of X-dosage in maybe somatic cells. So now I will come back to the project that I did, actually where I tried to understand what's the role of X-dosage here during embryonic development. So what we ask is double X-dosage, does it affect development somewhat in a sex-specific manner? So what we do, we used to study this mouse embryonic stem cells because they are derived, here from the inner cell mass of the blastocyst. And they're a very nice model to study effects X-dosage and axon activation, because the female ES cells, they will have 2 active X chromosomes, and when you induce them to differentiate, they will undergo random axon activation. Which you can see here in these RNA fish images. So what is stained here in green is the nascent transcript of an X-linked gene. So in ES cells, both copies of this gene are active, are transcribed. Then during differentiation, Xist starts to be upregulated and to code one of the 2 X chromosomes, and leading to gene silencing here. So what we started here in this project was that we compared differentiation of 3 different ES cell lines with different genotypes. So we have a female line with 2 X chromosomes, then we have a male line, an XY line, and we have this XO line, which is a subclone of this line, but has lost 1 X chromosome, but is otherwise identical to that cell line. So all 3 cell lines, they were differentiated towards epiblast-like cells, and at many different time points, we analyzed their transcriptome through microarrays back then. That's quite a while ago. So and this we chose to do this over 3.5 days, because that's the time window when axon activation is first initiated. So you see, Xist starts to be upregulated between day 1 and day 2 of differentiation. So when we did the kind of global comparison of the transcriptomes of these different cell lines, so in this plot, what we compare is XO cells versus XY cells. This heat map, the light color will indicate that overall gene expression similarity. So where it's light, the gene expression is very similar between different time points and the different cell lines. And the red line indicates a maximal similarity. So here the red line goes along the diagonal, which means that these 2 cell lines, they will differentiate with very similar kinetics. This looks quite different when we compare it to the XX cells here, because now this red line here deviates from the diagonal in a way that at day 3, the XX cells, they are most similar to day 2 in the XO cells, and that's very similar if you look at the XY cells. So this means that the XX cells, they're somewhat delayed in their differentiation compared to cells with just one X chromosome. Then we try to understand which genes are actually delayed in this. This is a transcriptome-wide analysis. So we somewhat quantified this delay and then performed a gene ontology analysis to understand what type of genes will be particularly delayed. And the main term that we found was stem cell maintenance. So these are genes, so-called pluripotency factors, mainly that are expressed highly in the stem cells and important to maintain the stem cell state, and then they will be downregulated during differentiation. So this is some examples of these genes. In our time course, so in red you have the XX, and you see that all these genes, they're maintained at much higher levels in the XX compared to the XY or the XO cells. Now to exclude that this effect we observed here was just something weird about this female line we looked at, we also analyzed another completely independent XX-XO cell line pair that was differentiated with different protocol in this case. And again, we observed that the XX cells maintain much higher levels of these factors. So this suggests that 2 X chromosomes seems to somewhat slow down here cell differentiation. So then we performed a large number of experiments to understand what's the kind of molecular basis for this. And to explain this, I give you some background on what is thought about ES cells and ES cell differentiation. So ES cells, they can essentially kind of reversibly transition between this so-called ground state of pluripotency and the primed state. So the ground state or naive state is where pluripotency factors, so these stem cell factors, they're very highly expressed. The transition between these is strongly dependent on the MAP kinase signaling pathway, which is generally associated with differentiation here, and will repress these pluripotency factors. And in addition, what is essentially a hallmark of the ground state compared to the primed state is that we have very low levels of global DNA methylation in these cells. And that's also somewhat controlled maybe by the MAP kinase pathway. So what we found now is actually that the presence of 2 X chromosomes will shift this balance towards the ground state here. So we first show you one example for pluripotency factors. So this is NANOG, it's a famous pluripotency factor, and this is generally expressed higher in the XX cells, which is the red, compared to the XO cells, which is the dark blue here. So the XX cells express higher levels of pluripotency factors, also of others. Then, so it's suggesting that dosage increases expression of these factors, potentially. Then we look at the MAP kinase signaling pathway. So first I show you data for a well-known target gene of MAP kinase signaling called EGR1, which in generally it's expressed higher in XO cells, shown here in blue, compared to the XX cells, suggesting that somewhat in the XO cells this pathway is more active. And we also look at this more globally, where we analyze large panels of target genes for different pathways. And so what is plotted here is the expression of these target genes in XX cells relative to XY cells in this case. When we focus here on the MAP kinase pathway, this one, in the gray, you see that this is, so generally XX cells express lower levels of these target genes of the MAP kinase pathway in these culture conditions here that are denoted with a serum, while in white, this is different culture conditions that block the MAP kinase pathway and actually are known to shift all cells to the ground state, so where you don't observe this anymore. So X-dosage, in addition, seems to reduce MAP kinase signaling. So finally, we look at these global DNA methylation levels here. This has been well known for actually quite a while, that at least in these cell lines here you have cells with 2 X chromosomes that only like about 25% global DNA methylation, while cells with 1 X chromosome have between 60 and 70, so it's a very big difference. So X-dosage also seems to reduce DNA methylation. So what we can say overall is that the XX cells, they exhibit increased levels of pluripotency factors, decreased activity of the MAP kinase pathway, and decreased levels of DNA methylation. So what we hypothesized from these observations is that there is actually one or maybe several genes that are located on the X chromosome that are present at higher dose in female cells. And that will somewhat maybe inhibit the MAP kinase pathway and thereby lead to these different phenotypes. So if this is the case, what we would predict if we can take a female cell and turn off one of its two X chromosomes, that this should reverse, it should make the female cells look more like male cells with respect to these different phenotypes. And so to do this, I established this cell line where it's a female cell line, where in front of one of the two Xist loci, you have a doxycycline-inducible promoter. So when we take these cells, treat them with doxycycline, they will upregulate Xist, like shown here by RNA fish, and lead to gene silencing of one of the two X chromosomes. So now we use the system to look at these different effects. So we start to look at signaling. Here you can focus on the MAC phosphorylation, that's part of the MAP kinase pathway. What is plotted here is in black, you have the DOX-treated XX cells relative to the untreated control. So you see that phosphorylation is reduced upon DOX treatment, and this effect is similar than if you compare XO to XX cells, which is in white. Now we look at pluripotency factors. They are also all reduced upon doxycycline treatment, making the female cells look more like male cells that have lower levels of pluripotency factors. We also see change in DNA methyltransferases. So these are the enzymes that are probably responsible for these differences in DNA methylation. They are increased upon doxycycline treatment, and similarly we observe a global increase of DNA methylation, making again the female cells look more like the XO or the male cells. So we can conclude from this that it's actually indeed X-dosage that shifts ES cells towards this pluripotent ground state by inhibiting the MAP kinase pathway. So now if our model is correct, that it's the double X-dosage that somewhat blocks transition here to the primed state and to differentiation, we would think that this block should be released during axon activation, because when one of the chromosomes gets silenced, dosage of this X-linked factor will be reduced, which should kind of allow the cells to differentiate. So first thing we did to test that, so yeah, what would be predicted from this is that if we can look at cells that have already done axon activation, they should be able to downregulate pluripotency factors compared to cells that maybe have not done axon activation yet. So for this, we did a single cell RT-qPCR back then, which allows us to distinguish, identify individual cells that either express Xist or don't express Xist, and then we compare expression levels for different pluripotency factors between these groups of cells. And what we see is that, particularly at day 4 of differentiation, so this is when axon activation actually has progressed quite a bit, we see that in gray here, the Xist negative cells will maintain higher levels of some of these pluripotency factors than compared to the Xist positive cells, suggesting that maybe it's actually these Xist positive cells that preferentially will downregulate pluripotency factors. However, this is just a kind of correlative evidence and doesn't say that these effects on pluripotency factors will be downstream of axon activation. So to identify a causal link, we tried essentially to alter the kinetics of axon activation and see whether this would affect the kinetics of differentiation. So we use again this inducible Xist system to accelerate axon activation, because when we treat the cells with doxycycline already 1 day before differentiation, Xist will be upregulated faster and to a higher level. And to do the inverse experiment, we used female Xist knockout, homozygous knockout cells, where Xist cannot be upregulated, so these cells cannot undergo axon activation. Now, when we look again at this panel of pluripotency factors. And what is shown here is the level with the DOX treatment compared to the untreated control. And we see that quite a number of these factors will be downregulated faster or to a stronger extent when we accelerate axon activation. And we see the inverse when we prevent axon activation. Here in particular at these late stages of differentiation, these cells, they maintain higher levels of pluripotency factors because they're somewhat still blocked in their differentiation. So we can really conclude that axon activation seems to require to kind of release this differentiation block in XX ES cells. So I'll summarize what I've shown you so far, where we essentially identified that somewhat inhibition of the MAP kinase signaling pathway by double X-dosage somewhat slows down differentiation of ES cells, and this axon activation is required to release that block. So what we think is happening here is that there's some kind of developmental checkpoint somewhat that makes sure that axon activation has taken place before a cell continues with differentiation, because axon activation is an essential process. So if the cells fail to dosage compensate, the embryo will die a few days later. And that something like this actually also happens in embryos had already been observed a really long time ago, where it was found that here right at these post-implantation stages, so right at the stages when axon activation is initiated, there is a developmental delay in XX embryos. And this is both comparing it to males, but also comparing it to XO embryos, suggesting that it's indeed X-dosage dependent. So what I just want to show you in the last slides is what we are doing now to understand what could be this factor here on the X chromosome that is responsible for this effect. So we are looking for X-linked genes that can decrease MAP kinase signaling, increase pluripotency factor expression, and delay ES cell differentiation. And to search for these type of factors, we set up a series of pooled lentiviral CRISPR screens. So what we do here, we take ES cells that express Cas9, and they're infected with a lentiviral guide RNA library targeting all genes on the X chromosome. So what we have now here is the pool of cells where each cell will knock out one gene on the X chromosome. And then we need to somewhat stain or have a reporter for the phenotype, I will go into in the next slide, and use that to kind of sort out cells that have the phenotype we're interested in. So for example, cells that have reduced or increased MAP kinase signaling. And then we use deep sequencing to identify the guide RNAs that were integrated into these cells in particular, and these will be genes that will induce that phenotype we're interested in. So we look at all these different phenotypes, all with some sort of these pooled screens. So we look at activity of MAP kinase signaling, which is reduced in XX cells. We look at pluripotency expression, in particular for NANOG expression, which is increased in XX cells. And we look at this for a differentiation delay. For this, we use this gene ESRRB, which is also a pluripotency factor, which is strongly delayed. Its downregulation is strongly delayed during differentiation. So to look at MAP kinase signaling, we first used a reporter, so a GFP under the promoter of a MAP kinase responsive promoter. But we also use staining to phospho-ERG and phospho-MAC in these screens. Then for NANOG, we have a cell line where NANOG is tagged with an cherry. And for the differentiation, we have a cell where ESRRB is tagged in a similar way. So we can measure all these different phenotypes by FACS and then sort them. And so what we did actually one big primary screen where we screened all X-linked genes. And for this, we just used this MAP kinase reporter. And then we took all the 100 hits from that primary screen and screened them for all these other phenotypes, for the phosphorylation levels, pluripotency factor, and differentiation. So this is a summary of the results. Each column is a gene now. Each row is a different population that we sorted in the different screens. So on top you have the MAP kinase reporter, for example. Here on the right is where you see the genes that do have some effects on our screen. They can be globally grouped. Here there's this top group here, which is red on top and blue on the bottom. These are all X-linked genes that have some kind of MAP kinase inhibiting phenotype. So these are the genes we are interested in. Then we also see a group of genes. These are autosomal control genes here that we put into the library to test our assay. And there are also a bunch of X-linked genes that have the opposite effect, the MAP kinase inhibitors. So we are interested mainly in this group of genes, and that we are currently kind of validating, so I cannot show you any kind of final results on this, but so the gene with the strongest phenotype, which is this one, it's DUSP9. This is a known phosphatase of ERG, so it's definitely an inhibitor of the MAP kinase pathway. But we think it actually acts together with some of the other genes, so that is actually a multi-gene phenotype we're looking at, but we can build now a much more detailed cartoon here where we say that this X-linked gene is actually DUSP9, which will dephosphorylate ERG, which is part of the MAP kinase pathway, and that's why you have reduced levels of pluripotency factors and reduced DNA methylation and differentiation. So with this, I just want to acknowledge the people. So most of the data I showed you, I did when I was a postdoc in Edda Schulz lab in Paris, in collaboration with Niels Blütgen and Johannes Meissing here in the Charité. Then I want to thank my lab at the MPI, in particular Oriana Ginolit, who did the screen that I talked about in the end. And yeah, and thank you for your interest.